bioinformatics, and biotechnology. Our commitment lies in providing world class technical and scientific education in fields such as bioinformatics, biotechnology, life sciences, and clinical research. Whether you are seeking hands on training, dissertation guidance, or distance based courses, BTI is your premier destination. With our corporate office located in New Delhi, we are strategically positioned to empower students and professionals to contribute effectively to research and development in the critical domain of bioinformatics and biotechnology. Our core center is on establishing strong foundational knowledge and elevating it to industrial levels. So, BTI thrives on innovation across a spectrum of disciplines, including bioinformatics, functional genomics, proteomics, comparative genomics, drug designing, vaccine designing, chemoinformatics, molecular biology and dynamics, and systems biology, among others. Our primary objective revolves around assisting and promoting training, research, and development in the realms of bioinformatics and biotechnology. Bioinformatics is the practical use of computational methods to analyze biological data, solving real-world problems in genomics, proteomics, drug discovery, clinical genetics, and more. It accelerates research, aid in personalized medicine, and facilitates drug development by extracting valuable insight from biological information. This interdisciplinary field plays a pivotal role in advancing our understanding of life sciences and improving healthcare. So, in vaccine design, bioinformatics help in predicting antigenic proteins, modeling immune responses, and selecting vaccine candidates. It helps expedite vaccine development, improve efficacy, and address emerging infectious diseases efficiently. So, proteomics and genomics provide extensive biological data, while bioinformatics offer tools to analyze and interpret this data, enabling insight into genes, proteins, pathways, and structures. Together, they drive advance in biology, medicine, and molecular understanding. So, in comparative genomics, bioinformatics facilitates the comparison of genomes across species, revealing evolutionary relationships and functional insights. Structural genomics help predicting the three-dimensional structure of proteins encoded by genes aids in understanding their functions and interactions. So, in both genomics and proteomics, bioinformatics accelerate research, enhances data interpretation, and facilitates a deeper understanding of biological processes, ultimately contributing to advance in medicine. So, bioinformatics enable the use of system biology to model, analyze, and simulate complex biological systems. It aids in understanding how genes, proteins, and molecules interact with cells, leading to applications in disease research, drug discovery, and personal medicine. So, under the guidance of Dr. Basha Javed Sir and his academic and industry experience of almost 10 years from various institutions, including Helix Infosystem Limited, Noida, Bioinformatics Institute of India, Indian Biological Science and Research Institute, SPS Educational Group, and now the Bioinformatics Technologies of India. BTI has achieved numerous milestones and some healthy collaborations over last 8 years with highly reputed institutes over time including NIT Noida, Pine Biotech USA, and IFTM University Moradabad. Now come on to research and training. So at Bioinformatics Technologies of India, we offer industry-leading bioinformatics training that caters to both beginners and seasoned professionals. Our mission is crystal clear, but to provide a platform for individuals, professionals, R&D organizations, pharmaceuticals, and software companies to harness the immense potential of Indian bioinformatics. You can visit the official website of BTI for further details about numerous research and training modules. Let me help you decide why you should choose the bioinformatics training. We at Bioinformatics Technologies of India, we provide you the extensive curriculum, hands-on experience, expert faculty, networking opportunities, flexible learning, and industry collaboration. So don't miss this opportunity to revolutionize the field of biology through bioinformatics. To enroll in our training program, today and embark on gearing up your journey of innovation and discovery. Unlock your potential and shape the future with bioinformatics training from Bioinformatics Technologies of India. So now, thank you so much everyone. So now without further any delay, let's begin with this workshop. Over to you, Tushar.
Thank you so much, Priyanka. I hope my screen is visible.
Yes, please.
Yes, yes.
Okay. Thank you so much. So, good afternoon, everyone. My name is Tushar Gupta. I'm the program executive at Bioinformatics Technologies of India. Today, I will be conducting a workshop on the topic Navigating the Genomics Landscape Insights into the Population Genetics with Bioinformatics. So, we will be starting with a very basic form. What is bioinformatics? But before that, let me tell you all the contents we will be covering in this workshop today. We will be starting with the introduction of bioinformatics because we need to establish the base. Then we will go to data and databases, discussing different stages of data, data curation, data normalization, types of databases. Then we will start our hands-on training with BLAST, differentially expressed gene analysis, and gene set enrichment analysis. So, what is bioinformatics? As you all know, bioinformatics is a multidisciplinary field which includes the application and techniques or information technology, IT, and computer science, including statistics of math to solve the biological problem. Bioinformatics has emerged main as the main industry in 21st century because of its high potential of solving the biological problems that would took a lot of years in the past. One of the main goals of bioinformatics is to develop computational methods for analyzing large-scale biological datasets. As you all know, datasets are the small results that have been achieved by numerous biotechnology researches. Bioinformatics has the capability to club all those datasets, all those results together, and give out a fruitful result in a very short span and a very cost-efficient manner. So, applications of bioinformatics. Sequence, the first and the foremost, sequence alignment. Bioinformatics helps us aligning two or more DNA, RNA, or protein sequences together to find the similarities and differences. It would be included in our workshop uh, in the coming session ahead. Gene finding, as you all know, what is a gene? A gene is encoded by codons. So there are numerous databases which have all the data that is being stored in the form of information. When we feed our data inside those databases, we could easily find the gene which is responsible and also identify which genes is responsible for which pathway and how it helps or how it helps to increase or decrease the effect. Phylogenetic analysis. Phylogenetic analysis is the analysis by which the organism that is present together, it could help us find the evolutionary relationship with all the other organisms that were present uh, in the past. Microarray data analysis. Microarray data analysis is helpful in identifying genes whether differentially expressed under different conditions. Protein structure predictions. Long chain of amino acids exist. How they club together, they form primary, secondary, tertiary, and quaternary structures. They might be very tedious in finding out by biotechnology in methods, but it is quite simple and very easy to find through three-dimensional structures by using bioinformatics. Drug discovery. As it was discussed before, drug discovery is a method which can be used to find a ligand for a specific protein, which could in the future be helpful to find a drug that could heal a disease which might not have a cure now. Data repository. Bioinformatics provides a database to feed the data inside it, which could be easily accessed by anyone all around the world. Different types of bioinformatics. There are numerous types of bioinformatics. I will be discussing some of the main ones, like sequence analysis. As I discussed before, it helps in analyzing the sequence of DNA, protein, or DNA, proteins, and RNAs. Structural biology helps to understand the structure of a given organism, of a given cell, organelle, etc. Systems biology is the study of biology in which the entire system of a organism is studied. Like it includes all the metabolism, proteins that is being taken place, that are all the secreted proteins, etc. Comparative genomics, it helps to compare the genes of an organism to either other organism of the same species or different species. Proteomics is a study of proteins. Similarly, metabolomics is a study of metabolites that are being either used or produced during the metabolism. Medicinal informatics. Medical informatics helps to club together the data which is being extracted through the medical results, like clinical research. These data create a data set which is used in, by informaticians to conduct information, like to study the population of a specific disease and a specific region and to compare it. Chemoinformatics is the analysis of chemicals that are being secreted in the organism. And bioinformatics education helps us to understand and develop different modules to teach the forthcoming generations about all the advantages and all the advances that are being happening in the bioinformatics daily. What are the differences between bioinformatics and classical biotechnology? You all might be similar. You all might be aware of that. But here I am discussing how is the bioinformatics better than classical biotechnology. As you know, bioinformatics has a global reach. The experiment which is being conducted here in India will be the same as it is accessible to the person sitting in US, which is not that easy in case of biotechnology. Speed and efficacy. Bioinformatics provide high speed and high efficacy to the research and also accelerates the output of the research. Accuracy and reproducibility. Reproducing a module in the bioinformatics is quite easy as compared to those of biotechnology. Cost effective, the software is the only investment that you need. Software and training. Only after that, you can easily access that software to create and to analyze all the information that is being provided to you. Now, data and databases. For bioinformatics, data acts like a soul. Data is a piece of information that is being used to conduct the research using the tools for analysis of research. As you know, when there is a research in anywhere in the world, it generates some results. It it has some findings. All those findings are is the data that is being used by bioinformatics to analyze and to compare different data as compared to the different regions, different diseases, and different species. Data may be in the form of different types, like symptoms of disease, measurement of any quantity. It can be a result of any test or experiment. It could also be a structural information, like the 2D and 2-dimensional and 3-dimensional structures of proteins. The data may be primary data or secondary data based on the type of data and all the alterations that are being done on the data. The publicly and highly accessible data for use in research is stored in the online repositories known as databases. These databases are highly accessible and may be used by anyone who is seeking any help in bioinformatics. So, what is data curation and data normalization? As you know, when the research is conducted, we have a pile of data. That pile of data has our raw data. It may also has some unused information like NA values, the values that are not of any type, zeros, noises, and errors. There could be many cases where you would find a, a change due to human error, which is easily detected by bioinformatics software and which could cause an error. So we need to separate all those NA values zeros and find the raw data, which is further called as secondary data. The secondary data is a form of data which has all the data of our need. But during bioinformatics, we have software which are very specific to the type of data that is being entered. So we need to filter all those raw data and find our curated data. The curated, sorry. The curated data is the data which is most useful for us and which is highly accepted by the bioinformatics software for conducting the research. Different types of databases. As I discussed before, there are types of databases like primary database, secondary database, and derived or composite database. Primary database is the database in which the information or results from the experiment derived are stored directly, like nucleotide sequence database. These are the types of database in which the sequence of nucleotides are stored. For example, GenBank, DDBJ, and EMBL. Then we have protein sequence databases. Protein sequence databases are the databases which stores the sequence of amino acids in the form of different readable formats of computer softwares. The database enlists Swissprot, PIR, GenPep. Now comes the secondary database. Secondary databases are the databases in which the data of primary database is curated and normalized and all the NA values have been removed. These types of data may be of two types, like protein structure database, when all the empty missing values are removed from the sequence, and we only have our data in the 3D or 2D format, like PDB, EMBI, MSD, and MMBD are the databases which stores this data. Then we have motif and domain databases. These are the databases which focus on the specific site of a protein that is functional and that is useful for producing the result that we need. Like ProCite, Logs, and COG are the databases which come under this category. Then we have derived and composite databases. These are the four types, like structured databases. Derived and composite databases are the databases which have the data of our need, like they are very specific to the type and application that we need, like structured database like SCOP and CATH. The gene expression database, as it is very clear with the name, it has the data of all the genes with their expression, like how the gene expresses when there is an encounter with the disease or and in the normal condition, like we call the healthy condition and wild type condition and the diseased condition. Three types of database exist, known as GEO, GXD, and MGED. Then metabolic pathway databases. There are databases which contain the pathways of all the metabolic processes that are happening in the organisms. Three types of databases include KEGG, PathDB, and EMP. Then we have specialized databases. These are the databases which stores the data in the form that could be directly extracted and put into the bioinformatics software to analyze the results. So, after the theoretical part, now we will start with the bioinformatics work from scratch. Before starting, I would like to introduce you to the four steps of bioinformatics problem solving process. Like it includes identifying the problem. Step first is to identify the problem, that which type of disease or which type of population are we studying. The data before finding the data, it is very important and very crucial to identify which type of data is needed and which type of data will be fed inside the software to find. Then after identifying the problem, the second step comes finding out where we could find the data. What is the type of data that we need to find the type of data required for achieving the required goals. The step 3 includes we have to find the database which contains the data in the format that we require. After identifying the database, the fourth and the final step is search, extract, and execute the data using various web-based and standalone tools. So, before starting the experiment one, is there any doubt? Anyone want to ask something? Is there any question? So I don't think there is any problem. So we will be starting with the basic local alignment search tool. I want you all to be parallel with me. I will be guiding you through the whole step. You please focus and understand. And if you are stuck at any step, you can stop me right there by unmuting the mic. So, experiment one, basic local alignment search tool. As it is very clear with the name, this searching tool helps us to find the local alignments of the sequences of nucleotide or protein sequence. You all might be familiar with the abbreviation name BLAST. So, what is a BLAST? The BLAST is an online tool used for the sequence alignment of any one type of nucleotide or protein sequence with the other type of nucleotide or protein sequence, respectively. BLAST allows us to understand how our sequence that we have is similar to any sequence that is stored in the database prior. BLAST has many applications. Like BLAST helps us to identify the unknown sequence from this large pool of databases. BLAST uses the sequence in the FASTA format, which could be easily derived from NCBI GenBank database. The work of BLAST is to identify all the sequences and give us the amount of similarity that they are similar. Like here, it shows 98% similar. Applications of BLAST. So, why we use BLAST? BLAST is said to be the most important software for a bioinformatician. At every stage, you will be using the BLAST due to its high application, like species identification. BLAST is BLAST can be easily used to identify a species from the stored pool of data. When we have an unknown sequence with us, we could easily run the BLAST and find out which it is similar to which of the following sequences or which of the following species. It could easily help us find the gene which the protein sequence codes. It helps to understand the phylogenetic analysis. Like if we have a sequence today and we want to find its ancestry, its evolution, how it was 1,000 years ago, that all the data is being stored in the BLAST software. We could easily enter our query sequence and find out how much similar it is to all the results that are being stored in the BLAST. Protein domain identification. BLAST is used to identify the domains of a large amino acid sequence. A domain is a part of a protein that encodes for the protein of interest. Primer designing. BLAST is highly useful in creating the primers, which are useful in the process of DNA amplification. SNP discovery, single nucleotide polymorphism, is the type of polymorphism in which it helps to identify which gene or amino acid has changed in the entire sequence. So we will be starting with the practical now. So this is the sequence we will be using. So I'm copying this sequence in the chat box. You all may copy this sequence directly into the NCBI and find. I will be sharing it here. We will be using the BLAST P. As you know, there are different types of BLAST, BLAST N, BLAST P. The BLAST N is used for the analysis of a nucleotide sequence, like, and BLAST P is used for the analysis of protein sequences. So here we'll be entering the section number of the query sequence. We can also enter the sequence in the FASTA format, GI or accession numbers like I did now. You can also align two sequences together. Here is the option of aligning two or more sequences. If you have two sequences and you want to find the similarity among them, you can easily do it using the BLAST. But now we will be using this accession number to find it into the large pool of database of BLAST. Here I have used the coronavirus surface protein sequence to identify how much which organism it is most similar to. Using the BLAST, it is very easy to compare. It is a very short sequence, so it should not take much longer time. But you should keep in mind that BLAST is a large software and it takes a lot of time. It refreshes itself. It requires a lot of time to work. So be sure you have a good connectivity of internet while working on BLAST. If there any question right now, you may ask till it is running. Are you all trying it with me? So it is still running. Thank you so much, Nilofer. So here we have got our results. So this is a job title that the BLAST gave to our sequence, query sequence program. The program that we run was BLAST P.
Sir, sir, actually, I didn't find that page. Can you please tell me again?
Pardon?
I didn't find that page. Um, near program also, I didn't get anything. So can you please tell me that before?
Can you please type? Your voice is breaking.
Okay.
BLAST page, can you please show me again? Yeah, sure. So this is the main official page of National Library of Medicine, BLAST. Like I told you, we have to know that which type of data we are using. In the example today, I picked up protein sequence of amino acids. So I will be using BLAST P, protein BLAST. So I have the accession number copied. I will paste the accession number here, Control V, and then we will be going ahead for our research. Like here, we will be selecting the BLAST P because we are using the amino acid sequence for the BLAST. Then we will click the BLAST and we will get the results. So this is a result that of the sequence that I used for surface glycoprotein of severe acute respiratory syndrome. You all may know SARS-CoV-2 COVID virus. So here it tells us the molecule type is amino acid. We all know we have taken protein sequence. Amino acids are the building block of proteins. Then the query length of 1238 amino acids. Here, this is the main table that we require. Here we see that similarity of the organism, of the different organisms. Here, this is a description. This is the scientific name of the organism from which we have find the similarity. The query sequence of 1238 amino acids that we took has been compared to number of sequences. Here we are displaying only 100, top 100. You could change the value to any value you want. Here the total score out of which we have calculated the sequence similarity is 2565, and we have found the 100% score with this one, surface glycoprotein. We all know we had took this one of severe acute syndrome. Then on the second we have beta coronavirus. With beta coronavirus, it has 100% score and the percent identity is 98.63%. Similarly, you can find it with the different types of coronaviruses. Horseshoe bat subcoronavirus, horseshoe bat subcovirus, rhizo. Their names might sound you similar, but they are different at genomic level. You can find the similarity. You can also find the accession number of all those. So you may compare and get a more detailed result of these. You can analyze any of these in your work because you all know that the sequence you have taken is similar, this much similar with all these organisms. Then there is a graphical summary. These red lines show the similarity. Our coronavirus is very general, so it has shown many red lines. But if you take a very less known one, it might not be this result. There might be different in with different colors and different similarities. This is the alignment. Like this is the sequence that we took, the query sequence, and this is a subject, the database sequence to which it compared. So here we can see that there is 100% similarity of 1238 amino acids. As we saw, 1238 was the total amount of amino acids that we had. So it was 100% similar to this one. You can also change it from here, next match and previous match, to compare how and at what point it was similar and at what point it was different. Like here you can see it was 99% similar. Out of 1238 amino acids, it was similar to 1221. And if you wish to find where it was different, you can easily find it from here. The homework sequence also there. That's nice. You all might be thinking how we are calculating this score. This score is purely based on the amount of similarity, differences, and gaps that are being encountered during the process of similarity. And here is a taxonomy. This is a type of file. This is a type of taxonomy that you may see from different lineage, organism, and taxonomy. It came from the root orthocoronavirus, beta coronavirus, SARS-CoV virus, and severe acute respiratory syndrome related coronaviruses. you can find how similar it was to different organisms of different groups. That's how you run the BLAST. You can also run it for nucleotide sequences, for BLAST, for amino acid sequences, for translated nucleotides, etc. Different types of BLAST have different application of running. So is it clear to all? Is there any doubt? Is there any doubt? Then we will go on second experiment. Gayatri, did you understand that I was explaining?
Yeah, I understood the solution part, sir, but I didn't understood that how to input that result because it's not showing in my laptop.
Okay, so let me do it. I'll show you run. You run the sequence on your end. Like here you can see enter query sequence. Is it visible?
Yes,
So here you
but
are. Yeah.
but in previously you said another very sequence, right? I mean, um, that one accession number.
Yes.
Okay,
No.
let me
Blast.
go.
Blast. I am telling you. Blast accepts the input in the form of accession numbers and GIs. You are thinking of accession number. What is it?
Yeah, we have to enter accession number, right?
You can find his accession number on NCBI.
Yeah, yeah, I got it, sir. Can you
Yeah.
please navigate me how to do again? We have to search, I mean, input again in job title. Job
Mm,
title.
like here I
Here,
enter the accession number.
yeah. Okay. Oh, yeah, yeah, got it,
Okay.
sir.
Yeah.
Now I get it. And then,
Then you don't have to worry about all these. These are the basics. When you will go ahead in your research, you can change all these parameters
okay,
like redundant. This is a database from which it is comparing. Here is non-redundant protein sequence. You can change all these
okay,
based on the research you are being conducting.
okay,
Like this is PDB, protein data bank. It contains
okay,
all the sequences in protein structures in 3D structure.
okay, sir.
Got it?
Yeah, got it.
Here's the program selection. You can select the type of blast you are using. Like for this, we have used amino acids, so we are using protein blast, blast
Yeah,
P. Okay, then you can click on blast, and you will get the results of this type.
okay, sir. Thank you.
You can try the sequence I gave in the chat box. Then you may reach me back. Okay.
Okay, yeah,
Is there any other doubts?
yeah. No, sir.
Thank you, Gayatri. So if there are not other doubts, we can go to our experiment two. Experiment two, differentially expressed gene analysis. So before understanding that, you should understand what is differential expression analysis. Differential expression is the expression of different features when the organism encounters any disease. Like when you are attacked by the virus, you get fever. This is a weather of fever. The fever is the response of the body against that virus. It increases the body temperature, which reduces the effect of virus. So this is a type of phenotype of our. This is a type of feature of our body. Similarly, there are other features. These features may include genes, transcripts, exons, and other different features. The goal, sorry, the goal of differential expression analysis is to compare the expression of two situations, like in the wild type when the organism is healthy and when the organism is diseased or it has encountered any type of disease. In our case today, we are comparing genes. So we will be focusing on gene expression. Our goal would be to understand how the gene works and how there is a change in the expression of genes once it encounters any disease. So expression value. You can easily understand with this graph. The yellow line which here is the difference that we will keep for separating the genes based on their expression. If on the encounter of disease, the expression of gene increases, then it becomes upregulated genes, which are here shown in the red. And when the normal expression of those genes reduces on encounter of disease, it is called downregulated genes, and these are here shown in the blue. Our main goal is to compare the means of this, the average, the red bar and the blue bar with the yellow bar, and how different they are to each other. When these three bars are not more far away, you may find no significant difference in those. I will, I will it will be more clear once we proceed ahead. Different factors of DEG analysis. Here is a fold change. Fold change is the main change of the expression of genes. When the gene encounters any foreign pathogen or any foreign stimulus, its expression changes. Either it increases its expression or it decreases its expression. Those change in the expression is called a fold change. It is represented in the logarithmic base 2 format. Then we have adjusted p-value. Adjusted p-value. P-value is the amount of probability that the gene changes itself. And the adjusted p-value is the recalculated part of that change value as compared to the whole population. In biological data, we mostly use p-value less than 0.05. Means if there is a slight difference of 5% statistically, then we will take it as a change. So we will be using this gene expression omnibus database for extracting the data for our experiment. So gene expression omnibus, what is this? It is a database for expression profiling and RNA methylation profiling managed by NCBA. GEO database has the data of high throughput sequencing genomic data, including microarray data and RNA-seq data. All these data are developed by the clinical researches that are being conducted at the hospitals. These are like clinical patients' data, which we take and compare in different populations. These data need to confer the minimum information about a microarray experiment format. This is the link of GEO. So let's start with the. It's, is it clear to all? Shall we start with the research?
Yes, sir.
Thank you. I will copy this sequence for you in the chat box so you may do it with your own. Will you also compare primer design? No, Gayatri, we will not be discussing primer design in this session. Maybe in the future we will be doing so. So this is the
Okay, sir. Thank you.
yes. This is the data set that we are going to use. So let's go to the GEO database. Here, gene expression omnibus. This is the database which we are going to search our data in. You can explore this database very easily from resources and how to page. It is very useful when you are conducting researches on microarray or RNA-seq data. So we enter our sequence here. Let's see what this sequence is. Here. When I enter the sequence, this is the result I got. The status. It is the publicly available data on 7th November 2020. The title of this is Expression Profile of Early and Mid-Secretory Human Endometrium Revealed by RNA-seq. So this implies that this is a RNA-seq data of recurrent implantation failure. What is recurrent implantation failure? Recurrent implantation failure is a disorder in which the mother is unable to conceive the baby despite n number of um, IVF IVF experiments. Experiment type is expression profile by high throughput screening. Summary. overall design, contributors. So when you are conducting the research, you have to download these files for your work. But since this is a workshop and we have very limited time, we will be analyzing this using the Geo2R application. Geo2R application is an application which is built by NCBI to run analysis on a very small set stretch of sequence. When you are conducting a research on a large data set, you have many samples, either you are comparing two disease, two organism, two species, then you may require coding for that. For small data set or just for a trial, you can use this Geo2R. So here you can see we have this data. This is the table. It has groups, IVF and healthy. So these healthy ones are the wild type that are available, that are not being that have not gone through any IVF, and these are the IVF samples. This is the extraction number, title, source name from the endometrium of gender, female, tissues, and different. I was telling about this, that there might be errors. I have seen many data sets in which it was a female reproductive organ disease and the gender was given male. So you have to take care of the data that is being input. You have to study the data completely. So firstly, we need to define groups. Let me do it again. So we are grouping these data because we need to compare those data, like I show you in the blue and red format. So we need to distinguish them based on a healthy and IVF so that it could also compare it. So firstly, we will be using healthy. We will be selecting all the healthy ones here. The data is collected from Spain, as it is mentioned here. Yeah. So we have done with our healthy samples. There are no other. Let me check once. No, there are no more healthy ones. So after selecting healthy from here, I will be clicking here. All these samples are colored, means they have been grouped to a healthy group. Now IVF. In IVF, we will be grouping all those samples which have undergone IVF. Here. So you can see the data is grouped in two bases, IVF and healthy. So now it is very clear for the software to understand that the ones in the IVF are the diseased or the IVF one, and the blue ones are the healthy ones. After doing this, we will be clicking analyze. It will take a little while. So let me tell you what is happening at the back end. At the back end, it is comparing, like we had the GEO dataset, which contains the information and the expression of all the genes before, like healthy, and when they are diseased, here IVF. So this GEO2R is comparing at the back end all the expressions of genes and trying to distinguish them based on their expressions. It will display the result in a very pictorial representation, which would be very easy to understand. It uses different coding language, like here it is using R language for this. Here is the code, but you need not to remember this because it is running at its own here. You can change the options here based on the research you are conducting. Like you can change the log fold to threshold. Here I am using 0. You can change it anywhere. Significant cutoff 0.05. As I mentioned, in biological research we use 0.05 or 5%. Script GO2R. Yeah, we have got the results. So there are these are the pictorial graphs which we can see here. This pictorial graph shows a result and a very clear picture of how these all genes were stored in the dataset that we used. There are too many datasets. There are too many of these genes. So discussing everyone. Here a volcano plot. In the volcano plot, it is log minus log 10p uh, versus log 2 fold chain. The blue ones here represent the down-regulated and the red ones represent up-regulated genes. You might not know which genes here are these. I will be discussing that ahead. Here's a volcano plot. Here is a mean difference plot. This is very useful for us, UMAP plot. Here you can see there are three clumps formed here. Remember, I told you that there are few genes which are not responsible for when there is encounter of any external stimulus, and some genes don't express themselves, either less or more. So these are those genes. They don't show much expression here. Then this is the second clump, which shows the difference. Like these are the genes which had a change on encounter of a stimulus. Similarly, here it is. So we here we know that we had 21,261 genes that was involved in the here that was involved in our data set, and out of those only 9,451 were those which shows any change of expression. So now you would understand why I was saying to normalize and curate the data, because see the difference, 9,451 and 21,000. The remaining ones are not of much interest for our experiment, but they would give just error. So you need to clear your data well before analyzing them. So here is a table. We will just download this table first. Till it is downloading, I'll discuss these. Is it downloading? Yes, it is. This is the box plot. It shows healthy versus IVF samples. It shows a normalized counts of these genes, these number of genes. These are the genes which are involved in healthy versus IVF. These do not come have all those genes which are not of our use. It is running. So these are other graphs that you can see. You can use all these for your research. Is it downloaded? Yes, it is downloaded. So this is the table that is being downloaded. It might not be very clear to you all. So I will be copying it into Excel. Here, a table is copied here. It contains all the genes. So we will be naming this sheet as all genes. Is it clear till here? Any
Yes, sir.
doubts? Any doubts, anyone?
Yeah, how to decide the threshold?
See, threshold is decided on two bases. Firstly, the type of data you are using. Because when the threshold you set is more or less, you won't get the data of, you won't get the number of genes that you require for the analysis. The threshold plays a very vital role. You have to apply permutation combinations to identify. Mostly we take 0 or 1, not more than that for that.
So which is good, 0 or 1?
It is 0 here. We have taken 0. By default it is
Okay.
0. For your convenience you can change it on the need you need for your experiment.
Okay. Thank you.
Thank you. So we have got all our genes. So let's see how many genes we have got. It's a number 21,262 genes that we have here. We have gene IDs, adjusted p-values, p-value of every gene, IFSC, SE, stats, log fold to change. This is the expression that I was talking about. This is the expression. As you can see, here it is 0, here it is 5. Means this gene, you can distinguish it like it is more than 0. So it is upregulated, while this is less than 0, so it is downregulated. So we need to separate all these. So we'll be copying it, renaming it. The work I'm doing here is on Excel, but when since it is a very small data set, plus I'm doing it just for teaching you all, but this all this work is done using the coding languages like R and Python, and for large data sets. This is a very small set of data set, so it would give us a convenient result. Yeah. So we will be separating all these. Since you have seen we have 21,000 genes, and for bioinformatics 21,262 is not a big that number. So I will be changing it. I will be sorting it down to remove all those values which are not of my interest. Like here I am taking only upregulated genes. So I will be applying the filter. You all should know one more thing. Like we use Go2R. Go2R gives us a normalized file. Means there are no zero, missing or NA values. We have already got the curated data as I showed you in the PPT. It is a curated data. So this is the advantage. It doesn't take much time for that. And we get a normalized file, and you have to first see that is your dataset you are using is running on this Go2R or not. So we will be applying the filter. Upregulated, we will be taking 1. If I would take 0, then the number of genes which I will be taking will get less, and it wouldn't give a good result. So I would be taking 1 here. It has separated, and now I have 874 genes. It has removed all the remaining genes. These are the upregulated genes. These are all greater than 1 and show higher expression. It means they are upregulated. Similarly, for this, I will be applying the filter on log fold to change, because the fold chain is the main change that happened when the gene encounters a stimulus. So since there I took positive 1, here I am taking -1. The genes which shows the expression less than -1 only will be remaining, less remaining all will be removed. So here, these are the genes which shows the down regulation. And how many genes we have? We have 483 genes. So this is the most important tables you have obtained for upregulated and downregulated genes. You will be using these in your research. You can use these upregulated and downregulated genes in different softwares, because they demand upregulated genes separately and downregulated genes separately. I hope it's clear to all. And Is there any doubt, anyone? You may ask. So let me tell you one more thing here. When you are working on the large data set, or also on this one, you have to remove some columns from here, because many columns are not of our use much, like gene IDs, IFSC, stats, and these descriptions. We don't need these. We are not removing it here, but you may remove it when you are conducting the research. And the most of our use is these symbols. These symbols are like the ID that are given to all the genes, and these will be useful for us when we are going to enter in an experiment 3 gene enrichment analysis. So is it clear till here?
Yes, sir.
Okay, good. Are you all noting the steps I'm doing here?
Yes, sir.
Good. So let's proceed ahead. Yeah. Through the geo dataset, we have chosen the dataset, analyze it on geo2r, and analyze the result. So our
Sir,
next
if you don't mind, can you please again tell me that how to select the data
means
in G?
selecting a data is selecting a data mostly depends on the type of experiment you are conducting. Like there are various types of data. You can use RNA-seq data, microarray data. This is only for genes, which I'm talking about. Like data could be of
Okay.
any type, like protein sequence, nucleotide sequence, protein structure. You have to search a specific database which contains the data of your need. That would help you to come to the result of the date of the experiment that you are conducting.
Okay, sir.
Okay, well, so next experiment, experiment 3, gene set enrichment analysis. So what is gene set enrichment analysis? You have obtained all the genes, upregulated and downregulated are separated also. But you need to know that how those genes which we have separated are useful, and at what position, at what pathway, metabolism they are being used. So this is done using the gene set enrichment analysis. So gene set enrichment analysis is used to identify the pathways, or the function enrichment analysis is a method to identify classes or genes or proteins that are overrepresented in a large set of genes. Means you don't have to find like if where this genes is using, how to identify this gene, which, like you have seen this data set contains 21,000 genes. You are not going to see each gene separately. So how will we do that? So here is Panther database. Panther database is the database with the full form you can understand, protein analysis through evolutionary relationships. So Panther database is the database which contains the proteins that are being formed by the genes. Every gene has the ability to produce proteins. So Panther is a database which contains the information that which gene at which position in which pathway is used, and at which it proteins which type of proteins. So we are going to use the Panther database now. This is the scheme we are going to use. Geo dataset, enter the gene name, select organism, analyze the result. Let's do it then. The website could be reached by PantherDB, Panther database. Yeah. So this is the official page of Panther database. You can see here it is asking the IDs of the genes. You can directly upload a file here. Like I mentioned, you need to remove some columns which are extra in your information. That you will understand when you will conduct the research, that which data or which column is not of your interest. Like description is for the researcher to understand, but it is not of any use for bioinformatics tools. So here we will be entering the IDs of the genes that we have obtained. Remember to copy all genes, like 21,000 genes, because in upregulated and downregulated, all the genes have been filtered out. So we will be using the gene symbol. These gene symbol, as I mentioned, are unique for every gene. 21,261 gene IDs are being used. Here it is mentioned as gene symbol, but remember these are gene IDs. You don't have to use these because these may be present already. Yeah. So enter IDs. I have entered all the IDs here. It is not. See, here I have entered all the 21,000 gene symbols. I have used ID list. You have to mention which type of data you have entered, like ID list, previously exported text search, workspace, Panther generic mapping, etc. Selecting organism. This is the data from Homo sapien. For analysis, we are going to use this functional classification viewed in graphical charts, and we are going to use bar chart because it is very easily understandable, and it will run. So what is happening at the backend? At the backend, Panther is comparing each gene ID to the vast pool of database which it has, and it will give us the result of where this specific gene is involved in the metabolism and all the other parameters which will be seen here. You need to be very careful with what type of data you are entering and what type of organism you are using, because each gene may have a different effect in different organisms. So here is the result. It is a very good result, as you can see. Like here from the table you can see different colors represent different type of genes that are there. Like if I click on this one, it is a translation regulation. So genes are involved in DNA binding transcription factor, as it presented here in purple. The red one shows transcription co-regulated activity. You can click on these and see the genes that are being involved here. So here you can see the molecular function. You can change the type of ontology you are using. Like here molecular function, you can also see biological processes, like biological processes in which all these genes are responsible. Here you can see biological regulation, cellular process, detoxification, growth, immune system. These gray ones are responsible in immune system, these ones. So you can easily see that this gene is involved in this biological process, and how and which genes are regulated by the encounter of disease. Like if the disease is specific to brain, it will target all the genes which are responsible for producing the proteins in the brain or any other part of the body. Cellular content component, cellular component like cellular anatomical entities is in red, protein containing complex is in blue. Protein classes, there are different classes of proteins, cell junction, chaperones, cytoskeleton proteins. All these genes are responsible in all these factors. Now comes the pathway. Pathway is most pathway could be easily studied when you are studying metabolic engineering, because you can easily target or trigger the specific pathway. Like here is glycolysis pathway. So the ones in the green are mostly available in the glycolysis. Here, these all genes will be responsible in performing the glycolysis in the body, breaking of carbohydrates. So I hope you all understand all this. You can perform all these comparisons by your own.
So can you please tell me again the pathway part?
The pathway part. Okay. See, There are different pathways that are and that are there in the human body or any other organism's body. These are all the pathways. When you are studying metabolic engineering or you are aiming to create a genome scale metabolic model, these pathways are very useful for you, because these pathways, if you are targeting a specific pathway, say this one, alanine biosynthesis, you are targeting alanine biosynthesis, you want to either increase its production or reduce its production. So from here you can see that which gene is involved in alanine biosynthesis, so that you can amplify its production or either reduce its activity so that the alanine production reduces. There are different types of.
Yeah, yeah, got it.
Got it.
Yes.
So this is all. You all can, you are always open to explore all these databases. You can, if you want, you can take the screenshot of the slides that I mentioned and you can explore all these. So here I, so we have done with the data sets. So now if you are thinking that we could do this for all the data sets, so sorry, you can't, because all these processes that are conducted here, these are for small data sets. When you are working on comparison basis or a large scale, you need to learn the languages like R, Python, etc., to conduct an analysis at more uh, publication acceptable format. Like for R package for large data set, in gene expression analysis, you have to use Lima package for microarray data and DSEC2 package for RNA-seq data. Like GO2R, which I conducted now, it was RNA-seq data. It is mentioned always on the top when you see which type of data it is. And for gene ontology, you can use cluster profiler. These all are the packages of R language which you can study, in our forthcoming art training program. So thank you all for your time and attention. Feel free to resolve any query. Hope you guys enjoyed it. That's all from my side.